Okay, thank you everyone for tuning in to hear my presentation. My name is Liliana Wolf. I'm a graduate student and researcher at Texas A&M University in the Department of Ecology and Conservation Biology. And today I'm going to be talking about my research, which focuses on modeling the spread of white nose syndrome fungus, Pseudogymnoastis destructans, in Texas and Mexican karst regions. So before we jump into caves and bats and white nose syndrome, let's cover a bit on the background of emerging infectious diseases in wildlife. So emerging infectious diseases in wildlife have been increasing in number and severity since the later part of the 20th century. And this is an issue that deserves serious attention because novel disease outbreaks have the potential to extirpate taxa that were previously of least conservation concern and exacerbate existing struggles for already imperiled taxa. And famous examples in the Americas include introduced West Nile encephalitis virus, which continues to cause massive die-offs in American birds, sea star wasting disease, which has caused mass mortalities of ochre sea stars in the Pacific Northwest, the deadly amphibian chytrid fungus, which is now a multi-continent epidemic, leading to huge declines in amphibian populations, and now white nose syndrome in North American bats, which is the research subject that I'm presenting on today. So this increase in the prevalence of wildlife diseases can be explained by a suite of causal factors. Firstly, humans and livestock are increasingly encroaching into wild areas and into contact with naive populations of wildlife that are susceptible to introduced diseases. Also, shifts in climate and weather patterns brought on by climate change extend the range of pathogens to intersect with new hosts. And this is the case with sea star wasting disease, which experiences spikes in infection with rising ocean temperatures. And lastly, increasing movement of people, livestock, and goods creates a conduit for pathogens to cross previous geographic barriers, such as oceans or mountain ranges. And this is the case with white nose syndrome. So what is white nose syndrome? I'm sure everyone attending the NSS conference is familiar with white nose syndrome, but let's review. White nose syndrome is a virulent fungal disease. It kills hibernating bats. The disease is caused by the fungal pathogen Pseudogymnoastis destructans. White nose syndrome kills by infecting bats while they are in hibernation and their immune system is off guard. The fungus necrotizes their tissues, which causes this intense irritation. This causes the bats to arouse frequently from hibernation. And the growth of this fungus concentrates in areas of the bats on their nose, ears, and muzzle. And this physical attribute is what gives the disease its name. So the frequent arousal of these bats from torpor depletes their fat stores that they've saved up for the winter, and it causes the bats to dehydrate and starve to death. And there are also instances of bats becoming so awake and hungry that they fly out into harsh winter conditions looking for food. This food, of course, doesn't exist as there's no insect activity in the dead of winter. And then these bats end up freezing to death at the entrance to caves, and that's shown in this photo from Pennsylvania. So the first documentation of white nose syndrome in North America was in a cave in upstate New York called Howe's Cave. And this during a survey in the winter of 2006, biologists entered this cave and they were met with this just shocking sight. There's piles of dead bats, a near total population collapse. And at that time, biologists had no idea what was going on. They named the disease white nose syndrome because dead and infected bats developed that white fungal growth on their nose. And in subsequent years, white nose syndrome radiated out across these adjacent karst systems, causing massive mortalities in resident hibernating bat populations. And so now we know that this disease is caused by the fungus, Pseudogymnoastis destructans, which I'm going to be calling PD from now on to save time. And the prevailing theory is that this fungus was introduced to North America from Europe, possibly on the shoes of some globe-trotting cavers who couldn't have known at that time that it's very important to disinfect your equipment if you're traveling large distances between caves. And so currently white nose syndrome is documented in 34 states in the US and 7 Canadian provinces. And the disease that causes, the fungus that causes the disease, Pseudogymnoastis destructans, this PD, is present in an additional 6 states in the United States. And so there's an important distinction between the fungus that causes the disease and the disease itself. So you can think about it as if, if you've got E. coli on your skin. This pathogen might be present, but you are not infected with E. coli. And so there's a difference between finding the fungus in the cave and seeing symptoms of white nose syndrome in the cave. And typically the fungus arrives and is documented before there are any documented signs of the actual disease, white nose syndrome. And so PD was first detected in Texas in 2017. And as of this year, caves in 21 counties have returned positive tests for fungal presence. And each year, this fungal presence moves further and further south through Texas. And for the first 3 years after fungal presence was detected, there were no reported white nose syndrome cases or mortalities. And we were hopeful in Texas that maybe the temperate winters in the state would keep white nose syndrome from developing. You know, maybe these cave bats overwintering in Texas either wouldn't enter into a deep enough state of torpor to be infected, and they would just live in this cave environment alongside this fungus without seeing any ill effects. But unfortunately, earlier this year, there was the first reported case of a white nose syndrome mortality in Texas, and this central county near Fredericksburg in Texas. And here in Myotis bellifer, a cave myotis was found dead from white nose syndrome. And the spread of PD in Texas was very concerning before, but after this first case of white nose syndrome, it's really upped the ante for this research on how PD and white nose syndrome behaves in Texas karst systems. So now we have some background on the history of white nose syndrome in North America. I'm going to jump into the first section of my presentation, which focuses on modeling the spread of Pseudogymnoastis destructans in Texas karst regions. So just to recap, we just learned about the history of white nose syndrome in North America. It's a deadly fungal disease. It's killing bats. It's moving south. The fungus has spread southward through Texas. We saw our first mortality this year in Texas. And with every emerging infectious disease, the questions are always the same. Where is it going to go next? And what is it going to do next? So in this first section, I aim to answer these questions for Texas. So the objective of this first section was to build a model of suitability for Texas karst systems to see what areas of these karst regions have caves that are best for supporting the growth of PD and therefore support these caves that can be nexuses of dispersal for the fungus. And the idea here is that if a cave becomes infected, bats moving between that cave and other caves in the region will act as dispersive vectors. And they'll be moving this pathogenic agent from a producing source or an infected cave, and they will infect other nearby caves. So different caves can be better or worse at supporting the growth of PD. And the best measurement of suitability within these caves is internal thermal microclimate. And so microclimate data is useful in a number of ways. Firstly, previous attempts to model regional suitability for PD have relied on an ecological niche modeling approach. And this is useful when little information is available, but it does not encompass microscale variation, which is necessary to understand fungal growth differences on a variable landscape. And secondly, incorporating this real microscale measurement in model development facilitates a bottom-up build, which is more reliable in terms of predictability of suitable conditions. So all in all, thermal microclimate is a very descriptive variable when modeling suitability for PD. And this is because PD is actually a bit temperamental. It's a cold adapted fungus. It can grow between 0 and 20 degrees Celsius, but it has a very narrow optimal range for growth, which is between just 12.5 and 15.8 degrees Celsius. And we know this because while it is very difficult to give Pseudogymnoastis destructans a formal education, it can be cultured. So I based the analysis of my data on previous lab experiments conducted on PD growth, and particularly this seminal paper, Veront and collaborators 2012. Their research found that temperature is the most significant factor in PD growth. And so I used the Briere-2 growth function from Veront's experiment to create a proxy scale for PD growth suitability. The assumption is that this growth curve in the laboratory experiment reflects growth conditions in cave environments. So using this, I was able to score each cave microclimate sample on a scale from 0, which would be least suitable, to 1, which is most suitable. And this created a unit of PD growth suitability for further analysis. So to understand the likeliness of PD making a cave a home in Texas, ELUSB2 data loggers were deployed in 52 caves in Texas. And so each data logger recorded temperature readings at 1-hour intervals. And for each location sampled, external factors were collected from open source data sets from NOAA and NASA websites. And specifically, I extracted data from each point for external temperature, elevation, precipitation, and of course, I already had latitude and longitude for each point from the time of deployment. So I used this fitted internal temporal suitability data from Texas to feed a GLS model. And this was used to explain variation of cave internal micro temperature as a function of these external variables shown here. And so this produced a predictive model to estimate the proportional contribution of each external variable to predict microtemperature. And so this model explained 43% of the variation within the system, and it found that all of these variables significantly explained variation within internal cave microtemperature. And so the next step after building this model that predicts internal cave microclimates on a landscape scale was to apply it to the extent of Texas karst regions. And then we could apply our BREER2 growth model to figure out which areas of Texas are most likely to have caves that are suitable for sustaining this growth of PD. So in order to do this, I generated a grid of 10,000 equidistant, evenly distributed points contained within the limits of Texas karst systems. Then I extracted external temperature, latitude, longitude, annual precipitation, lithology, and elevation from each of those points. And I used the microclimate fitted model to predict internal microclimatic temperature. And so the resulting figure on this slide shows from dark to light the areas of Texas karst which are predicted to be most likely to sustain thermal conditions suitable for the growth of P destructans. Okay. But of course, external conditions change seasonally. And so this figure was built using the same approach as the previous figure, but seasonally changing data, temperature and precipitation, were parsed out for each month of 2019. And you can see here that there are marked seasonal changes in predicting fungal growth suitability over the course of the year. And this makes sense when you consider that external temperature had the highest contribution to explaining variance within that internal cave microtemperature. So in conclusion, this first section showed that Texas karst has the ability to sustain thermal microclimate necessary for the growth of PD. And now this suitability for growth varies seasonally, but we can see that there are some regions that sustain this optimal condition for longer over the course of the year than others. And if we zoom into the summer months, we can better see these regions that maintain suitability for longer. So based on these findings, I would recommend that the northern areas of the gypsum karst panhandle, this northeastern section of the Edwards Plateau, and the Guadalupe Mountains area of West Texas be closely monitored or more closely monitored for the development of white nose syndrome, and that these areas be targets of interest if a treatment is developed in the future. This second section of my presentation explores suitability for growth of pseudogymnoasis destructans in varying regions of Mexican karst. So perhaps the most concerning thing about how PD is moving through Texas is the fact that 18 Tadarida brasiliensis, or Mexican free-tailed bats, have returned positive swabs for PD. Mexican free-tailed bats are the bats that you're probably most familiar with if you're like me and you live in Texas. They make up that famous bat colony under the Austin Congress Avenue Bridge. They're also the bats that, the species of bat that make up that enormous 20 million strong bat colony in Bracken Cave north of San Antonio. Well, these free-tailed bats have enormous migratory ranges from the US to as far south as Bolivia. And the major migratory route for free-tailed summering in Texas is between Texas and central Mexico, where these bats migrate along the Sierra Madre Oriental mountain range. So the idea here is that these bats could act as highly dispersive vectors for this fungus, introducing it from caves in Texas to caves in Mexico. And it's possible that if PD could gain a foothold in Mexico, whether it becomes virulent or not, these infected caves could become a nexus of dispersal, allowing the fungus to travel south until it possibly reaches areas in South America with harsher winters and a more vulnerable wintering bat population. So now that PD is present on free-tailed bats, we can expect that caves in Mexico will be exposed to the fungus. The question now is, when these spores are introduced to caves in Mexico, will these fungal spores just sort of sit impotently on the cave floor and exist in the same areas where bats exist? Or will they grow and sporulate, allowing these caves to become these sources of fungal dispersal? So this is the question that the second section of my presentation is going to set out to answer. Okay, so first, a word on Mexico. The country of Mexico contains a vast amount of biological and geographic diversity. Within the country, there are 7 ecoregions, including the North American deserts and temperate sierras. And a wide range in biomes has made Mexico a key spot for biodiversity conservation. It's estimated that Mexico contains somewhere between 10 and 12% of the world's biodiversity and approximately 11% of the world's mammals. Pertinent to this research, Mexico is home to 138 species of bats, approximately half of which are cave roosters. The karst regions in Mexico are concentrated along high elevation limestone and volcanic deposits in central mountainous regions, and also this coastal limestone deposit in the Yucatan Peninsula. So for the purposes of this study, 24 data loggers are deployed in 6 states and 2 mountain ranges: the Sierra Madre Oriental, which runs longitudinally through 6 states in the northeastern part of the country, and also the Eje Volcanico Transversal, which is an active volcanic belt which runs latitudinally through south central Mexico. The majority of caves in this survey were located from existing records provided by collaborating biologists. And a fun fact is that 2 of these caves sampled in Nuevo Leon were totally new to science. These were first documented during an expedition survey led by Peter Sprouse of Zara Environmental during the summer of 2019, and I got the opportunity to tag along on that expedition. This is very, very cool. But this is the part where I have to acknowledge the terrapus in the room. My research was possible because of a bat disease, but it was also impossible to complete because of another bat disease. And the original plan for this Mexico section involved a model of karst thermal suitability for Mexico, similar to the one I did for Texas in my first chapter. But for extraneous reasons, my collaborators and myself were unable to travel to the field this spring to retrieve equipment. So the bats giveth and the bats taketh away, but if they're able to behave themselves, hopefully I can get that data back later this summer and build the Mexico model. Fortunately, I did secure a grant to complete my fieldwork earlier this year from the Brufford Foundation, and they've been very understanding, and those funds will continue to be available once it's possible to complete my fieldwork. And with the coverage I had of currently deployed equipment in Mexico, I anticipate being able to develop a model equally as robust as the Texas model for Mexico. But for today, I'm going to share what I've learned from the 4 data loggers that I was able to retrieve before everything went to guano. So here are the locations of the 24 data loggers that were deployed to measure cave microclimate in Mexico. But unfortunately, due to the COVID-19 pandemic, only 4 of these data loggers were able to be recovered for the purposes of this presentation. But they all returned usable data, and so I will discuss them individually, and we'll also talk about the implications of their recordings. So the 4 data loggers I'm going to discuss today represent 2 different Mexican ecoregions: the American deserts and temperate sierras. And these ecoregions differ markedly in climate, precipitation, and elevation. Mexico 1 and 2 were deployed and collected from the municipality of Musquis, which is situated in the northern area of Coahuila State. Musquis is contained within the Coahuila Desert, which is hot and arid. The area doesn't see much rainfall, save for a monsoon season in the early summer, and then a few smaller rain events throughout the winter. All of the caves surveyed in Musquis for this project were formed from limestone karst and are generally accessible only from the tops of mesas, which are these isolated flat-topped landforms on otherwise flat surfaces. The vegetation in this region is mostly characterized by scrub brush and cactus. Mexico 3 and 4 were deployed and collected in the mountains east of the town of Zaragoza in southeastern Nuevo Leon state. These caves exist in the high elevation karst region of the Sierra Madre Oriental mountain range. This high elevation area experiences a relatively moderate climate. They also see a late summer monsoon season, but generally there are more frequent precipitation events throughout the year, especially compared to muskies. And this region is characterized by coniferous forests, oak forests, and agave. So these 4 data loggers were collected, and since I didn't have enough point data to generate a model of the karst regions in Mexico like I did for Texas, I instead applied this Briere-2 growth function from the Varant and collaborators paper directly to the data to get an idea of what we were dealing with in these caves. Mexico 1. So the existence of Mexico 1 was first documented during a caving survey expedition in Muskies in November of 2018. This cave is accessible from the top of a mesa, and it resembles a pit. It's a steep 50-meter drop with no branching tunnels. At the time of deployment, 1 bat, believed to be a Cornurinus townsendii, or Townsend's big-eared bat, was present. The data logger was deployed in this cave near that sighted bat, and that's about 30 meters from the entrance, and then the data logger was collected the following year. This chart in the bottom right-hand side of the slide shows the temperature variation in the cave over the course of the data logger deployment. The internal temperature of these vertical pit caves likely vary more in accordance with external temperature, since their rooms are more exposed to elements than, say, multi-room horizontal caves, like we're going to see in a couple slides. And so the recorded data from this cave returns a thermal suitability score of 0.79. Remember that that is on a proxy scale, from 0 being least suitable to 1 being most suitable. Okay. Mexico 2 was first documented during that same survey expedition as Mexico 1. This cave is also vertical and pit-like with an entrance on top of a mesa, and it was about 55 meters deep. There were 2 bats present at the time of deployment, also believed Cornurinus townsendii. The data logger was deployed near these bats, about 20 meters from the cave entrance. And the chart in the bottom right-hand side of the slide shows the temperature variation in the cave over the course of the data logger deployment. And you can see how temperature within this cave appears to vary in line with external temperatures, hotter in the summer months and cooler in the winter months. And the recorded data from this cave returns a thermal suitability score of 0.74 on that scale from 0 to 1. Okay. Mexico 3 exists in the mountains east of the town of Zaragoza in southeastern Nuevo León state. This cave region exists in the high elevation karst region of the Sierra Madre Oriental mountain range. And Mexico 3 is a horizontal limestone cave with 2 large rooms and a 3rd smaller, highly decorated side room. At the time of data logger deployment, there were 4 Cornurinus mexicana, which were present in the 1st and 2nd room of the cave. A data logger was deployed in that 2nd large room. And the chart in the bottom right-hand side here shows the temperature variation in the cave over the course of the data logger deployment. And you can see here that temperatures in this horizontal cave hold steadier for a longer period of time than the pit caves and muskies. These caves are multi-roomed and horizontal and more sheltered from the elements, which could explain how this the trend in temperature is a little more steady. And the recorded data from this cave returns a thermal suitability score of 0.85 on that interval between 0 and 1. So Mexico 4 is also in southeastern Nuevo León. Similar to Mexico 3, it's part of the high altitude karst region of the Sierra Madre Oriental mountain range. This is one of the caves that was first documented and surveyed during that expedition last summer. And so this data log was deployed as part of the initial cave survey. And you can see the resulting cave map from that survey drawn by surveyor Bev Shade. And this map details right where the data logger was deployed right here. So this was a 32-meter deep limestone cave with 2 steep drops. The data log was deployed in the bottom room of the cave near a pile of old guano, which suggested bat activity. And the chart in the bottom left-hand side of this slide shows the temperature variation in the cave over the course of the data logger deployment. Similar to Mexico 3, you can see this multi-room cave held steadier for a longer period of time than those pit caves and muskies. and then tapered lower in the winter months. And the recorded data from this cave returns a thermal suitability score of 0.84 on that scale from 0 to 1. So I hope to retrieve the rest of the data deployed in Mexico so I can finish building that Mexican model. And thankfully, I had the funds to do so through the Rufford Grant Foundation. But in the meantime, I think it's notable that all 4 of these caves surveyed so far in Mexico have returned pretty moderate scores for suitability for fungal growth. The karst area where Mexico 1 and 2 were deployed had slightly lower scores than 3 and 4. This may be explained by how the region where 1 and 2 were deployed in Muskies experiences higher average annual temperatures than that high elevation area where 3 and 4 were deployed. And since there is much higher elevational diversity in Mexico compared to Texas, it's possible elevation will explain more variation in cave thermal suitability for fungal growth when we're building this model for Mexican cave microclimate. And this is important because studies have shown that this steep karst in the Sierra Madre Oriental creates like a patchwork of elevation-specific suitability for flora and fauna. And this could support different fungal growth suitabilities between caves of different elevations, but say are otherwise share similar attributes or are spatially near each other. Also, it's important to note that Mexico 3 and 4 are in regions where there exist known migration colonies of Leptonicterus nivalis. This is an endangered species of nectar-feeding bat, and it's the principal pollinator for multiple species of agave. And the species is already imperiled by loss of habitat and anthropogenic disturbance. So if white nose syndrome were to develop in their colonies, it could have the potential to be a very serious issue. And in closing, this tool uses microclimate as a base variable for developing models to understand PD spread and can be very useful at targeting areas of concern on a landscape. In the future, I'd like to see this method deployed across all areas of the Americas where white nose syndrome might spread. In the early days of scrambling to prepare for and manage white nose syndrome spread, the only option was to take this blanket approach to surveys and check and swab as many caves as a biologist or land manager could get access to. And this is pretty tough on resources, and it's often difficult in areas like Texas and Mexico where many caves are either on private property or in areas that are difficult to access. So this tool has the potential to target areas of concern on a landscape and therefore offer recommendations that allow managers to save time and resources and offer a better and more streamlined response to combat the spread of white nose syndrome. But going off of that in the future, I just want to reiterate, I'd like to see this sampling scheme applied across the Americas and anywhere white nose syndrome could ostensibly develop. And these data loggers are relatively cheap. They're only about $80 a pop, and they're super user friendly. So it would be pretty cool if cavers were able to implement microclimate sampling as part of their surveys and then contribute to the building of, say it's like a super robust data set for internal cave microclimate. All right, so in closing, there can never be enough room on one slide to thank everyone who's helped me with this research. I want to acknowledge my committee and all of the cavers and academics who have helped me in locating caves and surveying the caves. Particularly, I want to thank the Texas Caving Grottos and also Bev Shade and Peter Sprouse for organizing that trip to Nuevo Leon. And with that, I will end my presentation. Thank you.